Good afternoon. My name is Bob Dorson. I'm Director for the Division of Pharmacology Toxicology Review in the Office of Generic Drugs, and today it's my pleasure to talk about the research opportunities that exists for N-nitrosamines in pharmaceuticals, and specifically focusing on the pharm tox area or the safety assessment area. I'd like to begin with a disclaimer. This presentation reflects my own views and should not be construed to represent FDA's views or policies. The presence of nitrosamines in drugs has presented a challenge since 2018 when NDMA was identified in a valsartan drug substance. Since then, various nitrosamines have been identified across various drug substances as well as drug products, with various root causes for their formation. Now, despite an extensive publication history, there are numerous data gaps that exist when it comes to root cause as well as mitigation of nitrosamine formation, as well as for their risk assessment. In my presentation, I'm considering conventional nitrosamines such as NDMA as well as NDEA, as well as the nitrosamine drug substance-related impurities such as N-nitrosovarenicline, and there's others out there as well. These need compound-specific risk assessments, and all of the data necessary to conduct the risk assessments for these compounds is not necessarily in the existing literature. What we do know is that nitrosamines as a class are potent rodent carcinogens and probable human carcinogens. We also know that they vary in potency and may form from various root causes, and therein lies an opportunity for multiple areas of further research. First, further work is needed on the detection and quantification of nitrosamines using sensitive analytical methods. Further work is needed on the risk factors underlying nitrosamine formation, as well as the strategies to mitigate their formation in drug products. These first two sub-bullets are going to be topics that my co-presenters will be discussing in their presentations. My focus is more so on the safety assessment side, including that further work is needed on the endogenous as well as exogenous exposure to nitrosamines, as well as their metabolism. In addition, we need further research into assessments of mutagenic and carcinogenic potency of nitrosamines. Specifically, various conditions for experiments that are aiming to characterize their mutagenic potential, what battery of studies is necessary to characterize that mutagenicity, and then the potential for methods that might classify nitrosamines with regard to carcinogenic potency, and lastly, predictive models for mutagenicity as well as carcinogenicity. Ultimately, further research and collaboration on the chemistry and drug formulation areas is essential, as well as for the risk assessment of nitrosamines. This will be absolutely critical to advancing generic drug development and ensuring continued access to safe and high-quality medicines. The FDA held a nitrosamine workshop, including a panel of nitrosamine subject matter experts, so that we could better understand the current research for nitrosamines. And they highlighted that our exposure to nitrosamines is one area that needs further research, because there's a knowledge gap when it comes to endogenous formation or formation of nitrosamines within the body. Specifically, we don't know which nitrosamines are being formed and at what levels, and this really prevents comparison with levels that are detected in drugs. They noted that endogenous formation is difficult to characterize. For example, rapid metabolism complicates the quantitative determination of nitrosamine levels. There's an unclear rate and fate of distribution and excretion of intermediate metabolites to help inform nitrosamine exposure. We have minimal information on tissue and organ metabolism, other than that in the liver, which is relatively well characterized. In addition, there are still uncertainties over DNA repair capacity, and we have a general lack of knowledge on the sources of various adducts and the contributions of exogenous and endogenous formation of nitrosamines to those adducts. Exposure to exogenous nitrosamines from, for example, food or water is an additional risk, and that needs further investigation. In addition, the potential for biotransformation of nitrosamines can also inform their risk for mutagenic potential. However, we need further data to inform whether a nitrosamine will be metabolized. We need the data, so ultimately we can form models that will predict biotransformation of nitrosamines in the future. The evaluation of mutagenic potential of nitrosamines is another area that warrants further research. After root cause analysis and attempts to mitigate the formation of nitrosamines, sometimes there is a nitrosamine that warrants further safety assessment, and in that case, mutagenicity is a first step in nitrosamine hazard identification. So although many nitrosamines are mutagenic carcinogens, it's understood that they vary in potency, and in fact that some nitrosamines may not be mutagenic or carcinogenic. We need further research to characterize mutagenicity using current standards for testing, sometimes some different conditions, and in particular because there are these data poor NDSRIs for which there's not published literature, but for which we are looking to generate data so that we can better understand the mutagenic and carcinogenic risk of these compounds. So we were interested in an optimized testing battery to evaluate nitrosamine mutagenicity, particularly when trying to establish that a compound is non-mutagenic. So a standard bacterial reverse mutation assay, or an Ames assay, can be informative to identify the mutagenic potential of nitrosamines. In fact, many nitrosamines are positive in a standard Ames test. And in those cases, further identification of their mutagenic potential is not really warranted. However, if it's negative in the standard Ames, that's not necessarily sufficient to conclude that a compound is lacking mutagenic potential. And that's in part because literature suggests that there's species-specific differences in the metabolic activation of potential mutagens, such as nitrosamines, in the Ames assay. So there's current and planned research efforts at FDA to investigate the mutagenicity evaluation in the Ames assay, as well as in other in vitro and in vivo systems, some involving mammalian cells as well as in the TGR model. And so the mutagenicity evaluation is something that warrants further research, and it's also noted that there are collaborations among various scientific groups looking at this, and they're collectively looking to add to the overall body of work that will ultimately help to inform the mutagenicity, the reactivity, as well as inform the carcinogenic potency of nitrosamines. Computational toxicology is another area warranting further research. Nitrosamines vary in their mutagenic and carcinogenic potency. However, if compound-specific data isn't robust enough to derive an acceptable intake limit, then a read-across approach is really critical to identify a more structurally relevant reference compound so that we can determine an acceptable intake. Now, FDA's computational toxicology subject matter experts look at the chemical environment around the nitrosamine structural alert as an important factor when selecting an appropriate reference compound. And there are several research areas that remain to be explored, including structure-activity relationships to identify structural features that are mitigating versus activating for mutagenic and carcinogenic activity, structural methods to assign nitrosamines to a potency class, local similarity calculations of a nitrosamine environment, QSAR models to quantitatively predict TD50s, as well as models for prediction of metabolism. While progress has been made on the risk assessment of nitrosamines in drugs, there are several areas that need further research. Specifically, I'd like to highlight the Ames assay conditions and follow-on in vitro and in vivo assays as important aspects warranting further consideration. In addition, on the computational toxicology side, models for identifying reference compounds for predicting carcinogenic potency and a potential for classifying nitrosamines also warrants further work. Research on methods to mitigate the nitrosamine formation, as well as streamlining the safety assessment of nitrosamines, is absolutely key to advancing generic drug development. We very much view collaboration with international regulators as well as our counterparts in industry as a key to advancing this very important work. I'd like to finish by thanking the organizers of this workshop as well as the moderators of this important session. I'd also like to thank each of you for your time and attention. I very much look forward to the panel discussion. Thank you.